Welcome to you all for the distinguished lecture series today. And this session will be recorded, I'm required to say, so please be advised to say only what you want to say. But it's a great pleasure to welcome our lecturer today, Julia Schulze-Hentrich. And I was really intrigued to read something about you, and I have to get my notes about this, because we have a very special guest today, because she not only studied biology as a diploma, but she's also a teacher, or studied to be a teacher of biology and chemistry. Mm-hmm. This teaching, I think you have two hearts in your body if you want to teach, and this is, let's say, a wonderful thing for a university professor. So I noted that quite well. And you studied in Göttingen, Jena, but also in Berkeley and Yokohama. And I haven't been to Yokohama, so it's high on the list. So you have an advantage already. That's very nice. And your PhD thesis, I think you did in Vancouver. So you have traveled and seen the world, which is very, very nice. You also did your postdoc in Vancouver, and then you did your habilitation, your further career in Tübingen. So you know not only the world, but also Germany. And then you were promoted to be the full professor and lead of the Institute of Human Genetics in the Saarland, I don't know how you say that in
Saarland
English.
University.
Saarland University in 2023. So that's a good, very nice and wonderful career. You are a person of genetics and epigenetics with a favor of neurogenetics, and this is why we have not only the people of the Collaborative Research Center Sexdiversity here, but also the people from neurogenetics. Welcome, of course. And now I have to translate that, because when I Googled you, Google is always, it just strips you off, but it says something, your topic is the interplay of genetic predisposition, sex and gender, and environmental influences in the context of neurodegenerative illnesses. I hope I translated that right. And that's a very encompassing and interesting topic, especially for an endocrinologist like me, when you talk about sex and gender. So I'm very intrigued to hear your talk today. And just 2 days ago, your recent publication was put online in PubMed. So 2 days ago, that's on the 5th of May, it's on advances of single-cell transcriptome profiling in Parkinson's disease. So I think this is something you will tell us now, so we look forward to your presentation. Thank you very much.
Thank you so much for the very kind introduction, and also for the invitation. I'm really, really happy to be here to give a lecture today. Thanks for the organization, and also to making my stay so nice and lovely with this great weather. It's my first time in Lübeck, so I'm really excited to be here to learn more about your SFB. Myself, I just got into the topic of sex differences. I studied genetics and epigenetics, with a focus first on molecular mechanisms, how crosstalk between modifications really shape gene regulation, and then moved into Parkinson's disease and other neurodegenerative diseases during my postdoc and time in Tübingen. And I'm now at the genetics department in Saarbrücken at Saarland University. And today I would like to give you insight into 3 short stories we have been working on and are working on, but first start with an introduction on sex differences overall and how they also play a key role in neurodegenerative diseases. So I don't have to tell you in this audience that the scientific and societal importance of sex differences have been largely underestimated differences here exist as well, for example, on an anatomical level and also on the level of connectivity. And in this lecture series, you heard already all about the idea of also having a gender mosaic in the brain by Daphne Joel, who really nicely came up with this theme and clearly shows that it's not a black and white difference. It's very small changes that set us apart and that we need to study very carefully in a cell type and cell also context-specific manner to really understand how we come to a difference in disease susceptibility for several of these neurological disorders. We know that, for example, Alzheimer's disease occurs more often in females, whereas Parkinson's disease overall occurs more often in males. We know these correlations, but we don't understand the molecular underpinnings. So this is where we need to molecularly understand better, and then also at the end, hopefully treat better and diagnose better. And it all goes back to the central question of sexual differentiation. What are actually the inherent factors that does each sex have that make it different from each other? And there are many, I could give, I'm sure many of you could have several aspects added to this topic. Today in this presentation I will be mainly focusing on sex as a biological variable to really understand this key question here. And as a geneticist, of course, first we need to look at the sex chromosomes. Males and females, men and women differ with the composition of their genome, females having 2 X chromosomes, males having an X and a Y chromosome. But if you look at these differences, what also becomes apparent is that the X chromosome is way larger than the Y chromosome. The X chromosome has more than 800 genes. The Y chromosome, with less than 80 genes, has less genetic information, but key information that determine sex and that sets the both sexes also apart. And then there's this simplified view that you can take if you have these differences now between females and males. If you look in the literature, a strong focus has been put on the SRY gene as the testis determining factor that then leads to testis development and this peak of testosterone prenatally in animals and also in human that then already at this early time point shape brain connectivity and structure that is later on reactivated in puberty when hormonal levels rise again. But this overview is super simplified and neglects the point that there are many other factors, and also hormonal levels are not as flat in females as we can see it here. Early on already, there are both sexes actually produce gonadal hormones that make each sex different from each other. And some of you in this room really contributed or are contributing to this context and make a point to consider hormonal production in both sexes to really understand what sets the biological sexes apart. If you look at gene expression, it's clear that there are many differences beyond the Y chromosome. This is a study already from 6 years ago where detects data from almost 70 tissues have been analyzed, and sex-biased transcriptional regulation and gene expression has been investigated, and it has been shown that beyond the sex chromosomes, also all the autosomes show sex-specific gene expression and regulation, and we don't really understand where this comes from and what drives these differences early on, but also throughout the life. And me as an epigeneticist, I believe that epigenetics, of course, plays a major role, because this is the level where on top of our genome information is stored long-term, and where sex-specific transcriptional signatures are produced internally, because we have the epigenome is the programmatic of individual cell types. I'll come back to that later, but also by external influences or internal influences like hormones. These signatures are shaped and stored long-term in a cell type specific manner. And this regulation is super complex. There's not the one epigenome, but you have multiple layers of information. You have on the one end DNA methylation with chemical modifications added directly to the DNA, but also many post-translational modifications of histones, so the proteins where DNA is wrapped around, that can be added or removed and contribute to accessibility of the underlying DNA that is wrapped tightly in the tiny cell nucleus, and thereby influencing gene activity. We really have to understand also the crosstalk between all these levels, how they then enable gene activity to be silenced or to be activated. And in the literature, a strong focus is put on DNA methylation. There are many studies how this epigenetic mark really contributes to cell identity, but also is influenced by external factors and hormonal influences. It's mainly found in the context of CPGs, so cytosines that are followed by a guanine. They have this CPG structure with many CPGs in a row in these so-called CPG islands that are found in promoter regions of many genes. And it's kind of a silencing mark in promoter regions. The more methylation you have, the less the gene is expressed. And it's an easy trigger basically to turn genes on or off. But this is only one level. Again, post-translational modifications are widespread and very complicated. When it comes, lysines are amino acids that can be modified. And already if you just look at methylation of lysines, you can see that you have different cardinalities, mono, di, or trimethylation with 1, 2, or 3 CH3 groups that can be added, and it makes a difference if there's 1, 2, or 3 groups. And we have many lysines in these histones, in the many nucleosomes we have in our genome, and we still need good profiling and very clean data to really understand where we have these marks present in combination with also DNA modification marks to see how they influence gene activity of the underlying genes. Plus, as I mentioned before, the core of epigenetics is that these modifications really determine our cell type identity in the process of differentiation. In the middle here you see this famous picture of Conrad Waddington, the godfather of epigenetics, who termed this landscape of the epigenome, of epigenetics. You see a cell on top of a hill here that when it differentiates, it basically rolls down and has to take decisions into which cell types it will differentiate. And in this process, epigenetic marks are put on DNA and on the chromatin and are stable, a lifelong process to keep our cell types also in the proper state and regulate very tightly the genes that are necessary for many of these functions. But to come back to sex differences, the epigenome is also really important, for example, for X chromosome inactivation. In females, because we have 2 X chromosomes, we need to silence one of them to have gene dosage balance. Males only have 1 X chromosome typically, and when females have 2, one of these X chromosomes are silenced, and then they form a so-called bar body. If you look under the microscope, you can see that the silenced X chromosome is really highly condensed and thereby not accessible for regulatory factors. Genes are almost completely shut off. And this process, it's also important to note, does not occur early on in the zygote already, but the chromosome X inactivation only occurs in the 1000 cell state, randomly. So you have cells, as a female, we have cells in our body where one or the other X chromosome is inactivated. And you can nicely see that in these so-called calico cats. Only female cats have this pattern of orange and black fur, because here the fur color is encoded on the X chromosome, and you can see that either one or the other X chromosome is silenced. So as females also, we are mosaic for X chromosome inactivation, and not in all cells the same X chromosome is inactive. Plus, the simple view of complete inactivation has turned out to not be true. We see quite a bit of X chromosome escape that is also dependent on age, for example. It increases with age. More and more X-linked genes get reactivated and then are highly expressed in females. And you can see here that across many tissues and cell types, you can see a higher expression of genes on the X chromosome in females than in males. It's different for some of the genes that here are at this pseudo region where you have sequence similarities between Y and X chromosome. That's the other way around, but typically you have higher expression than from the X chromosome, also contributing to sex differences. So you can see it's not one gene that makes a difference, but it's many genes that get reactivated. Many of these genes are linked to immune functions, explaining also why some of the autoimmune diseases have different prevalences in males and females, and explain phenotypes we see there. Also on the Y chromosome, there are many more genes that contribute to sex differences. It's not everything is about SRY. There are many others that play a role and are also dependent on age, up or down regulated. Some of them are linked to epigenetics. Here we have a lysine demethylase that removes these methyl groups from lysines I just introduced, and directly then have an impact also on gene expression of several genes. So to summarize this first part, the current view also that Arthur Arnold, one of the experts in this field, just presented last week at the EMBL conference for sex differences of health and disease, showed that we have, of course, genetic factors that determine our sex. The Y and the X chromosome are here really key players. They are important to form the gonads. The hormonal effects, both on female and male sides, are important to shape sex differences also in non-gonadal cells. But on top of that, we have other effects we need to consider. There are also sex chromosome effects beyond hormonal regulation that shape our epigenomes in each individual cell that we need to understand to really come to conclusions why certain disorders like Parkinson's disease occur more often in males and females. This is now where I'm back at the topic of Parkinson's disease. Parkinson's disease is a highly multifactorial disorder affecting millions of people worldwide. Tremendous amount of work has been done to identify genetics of Parkinson's disease. Also here in Lübeck, key contributions have been made to understand genes that are involved in Parkinson's disease, and we know more than 100 variants today that are linked genetically, but they can only explain about 15 to 20% of all cases so far. The majority of cases is seemingly sporadic, have a most likely a genetic component, but are also influenced by, of course, age. Age is the key risk factor for Parkinson's disease, but also by environmental influences. There are risk factors for Parkinson's disease like exposure to pesticides or head trauma, but you also have protective factors like exercise or coffee drinking or smoking that have been correlated positively with Parkinson's disease. But most of these are only correlations, and we don't understand on a functional level how they interact with the genome, and these gene-environment interactions on genetic and epigenetic level are really important to further disentangle in a sex-specific manner, because with all the differences I just showed you on sex chromosomes and beyond, we have to really understand for each sex separately how these influences come together. And then you can look at sex-specific risk factors at different stages. This is a figure I took from a review from Enrico Glab from Luxembourg, where he summarizes that, of course, you can have sex-specific risk factor exposure, you can have sex-specific vulnerability, manifestation, treatment responses, progression. So there are also many levels and layers here where we carefully need to look in the course of the disease progression where sex actually plays a role as a biological variable. And if we assume that the genetic risk for PD is similar at the beginning of life, and then it shapes over time, one can envision that by sexual differentiation of the brain and the hormonal changes we see during puberty, menopause, before and after menopause, we can have maybe an influence on the risk to develop Parkinson's disease, but we also need to consider gender-biased lifestyle factors and sexually dimorphic responses. We know that men work more often as farmers and are exposed more often to pesticides. So maybe this is already a simple explanation why it occurs more often in males, because they are just exposed more to pesticides. But this is also too simple of a view. So we really need to understand the interplay of all these factors to really come to conclusions why we see clinically these sex differences in Parkinson's disease. And they are really clear. As I said, men are more affected than women. They have greater overall motor impairment, typical non-motor symptoms of cognitive impairment and sexual dysfunction, comorbidities like dementia, and also when we look at progression, faster decline of motor symptoms and cognition. Whereas female, when they are affected, women when they are affected have atypical motor symptoms, mostly dominant resting tremor, other non-motor symptoms like sleep dysfunction and pain, so other comorbidities like osteoporosis, and again also other treatment responses, higher risk of levodopa, dyskinesia, and so on. So to look at these differences, or to link these phenotypic differences to a molecular level is really challenging in human cohorts. You need large cohorts, you need really good metadata to disentangle all these effects. Or what we also do, you can go to animal models, for example, and I will show you now one study from animal models and two studies from human cohorts that we are doing and did to understand these sex differences in Parkinson's disease. This was a study we still performed in Tübingen at the Human Genetics Department with Olaf Ries, where we took a mouse model of Parkinson's disease, which overexpresses the human SNCA, one of the key genes encoding alpha-synuclein, which shows a clear phenotype of Parkinson's disease with age. And we housed these transgenic animals along with wild-type animals that didn't have this mutation, either in a standard cage. On the right here, you can see how a standard cage looks like. We have about 3 to 4 animals per cage, some nesting material, or we housed the animals in a so-called enriched environment. And this has 3 components. An enriched environment has components of more social interactions, so the animals were in a larger cohort, 8 animals instead of just 3 or 4. It has components of more physical activity, so more running wheels and toys, and so cognitive stimulation was achieved by rearranging these toys every week for an entire year. So we housed these animals in a standard cage or in these environmental enriched cases throughout their life, and observed that the phenotype it delayed the phenotype of the animals, the cognitive and the motor phenotype, and it also led to dramatic changes in gene expression. And I'm only showing you here one heat map we generated from RNA-seq data. So these are gene expression data where you look at the expression of the entire, of all the genes in a cell. This is bulk data, so it's a mixture of different cell types in the hippocampus of these animals. And we have these 4 groups: wild type, standard environment, enriched environment, then our transgenic animals overexpressing human alpha-synuclein, in a standard cage or in the environmental enrichment. And red and blue just means up or down regulation of genes when you compare them to the wild type standard animals. And I hope you can appreciate that over time, when the animals are here 12 months old, you can see that the transgenic animals show differences compared to the wild types. Genes that are up or down regulated. Each row here represents one gene, and each column is one animal. So all 4 animals in this group showed up or down regulation of about 500 genes. But if these animals, the transgenic, their siblings basically, have been housed in environmental enrichment, you largely prevent these molecular disturbances, and the profile looks more like a wild-type animal. And you see this not only for neuronal genes, you also see this for other glia cell types, astrocytes, microglia, oligodendrocytes, across the data set, indicating that this protective effect is not just neuronal, but also occurs for the glia cells that support the neurons in this context. And then, of course, we were interested to understand what drives this protection. You can then ask what are the factors that particularly go up in the transgenic animals that were housed in the environmental enrichment. And we could identify several so-called immediate early genes that are also linked with cognitive stimulation. They go clearly up when the animals have access to more exercise and a larger cohort. And we could see that on protein level, this increase of transcription led also to balancing out of the effects we saw reduced in these transgenic animals that were housed in the standard cage. And then you can ask for all these regulators in which cell types are they found. And for each of the cell types, we could basically attribute one of these immediate early genes, with EGR1 playing a key role because this is a gene really upstream of this regulatory process. And EGR1 is a sex-specific regulator of neuronal chromatin and other synaptic plasticity effects. What I haven't told you yet, we did the study in female animals. In contrast to other experiments that are typically done with male animals, here the vets did not allow us to do this experiment with male animals. They told us you will not be able to house 8 mice in a cage, 8 male mice in a cage for an entire year without them killing each other or having a strong hierarchy leading to a separation of these animals. So we only have data here for females, but we are now planning to rerun this analysis with males that are housed in environmental enrichment for shorter time frames. Because we really, if we want to understand sex differences, we need to see how males react to environmental enrichment, and if the sex-specific regulator for both biological sexes really comes to the same effect. But this also shows you that it's not trivial sometimes, even in animal research, to look at both sexes, because the paradigms do not work out equally well for males and females. The next study I wanted to show you is a study we've performed together in a consortium called DIGPD with colleagues from France and from Canada, where we used large cohorts from our French colleagues, Alexis Elba and Alexis Brice, who have, for example, the TER cohort, the Parkinson cohort that also includes wine farmers and have a nice documentation of their pesticide exposure, and genetics has been done on these patients, on the blood samples that are available. We used the blood samples of these patients and controls to, on top of the genetic profiling, also look at DNA methylation differences, so epigenetic differences that occur in blood samples of these patients and as well as controls. And the key aim here was also to disentangle genetic effects from environmental effects and see if what is driven clearly by genetics, what is driven clearly by environmental influences, and where you also have interactions of gene and environment and additional effects of the interactions then on top of that. And when we just fused our data and used sex as a biological variable in our model to find differences, we found nothing. There were no CpG DNA methylation differences that we could find between controls and Parkinson patients. We only found differences when we stratified our analyses and had the females and the males analyzed separately. Then we actually found more differences here in females on DNA methylation level compared to males, indicating that you really need to do at least the epigenetic analyses in a sex stratified manner to find these changes we can that are linked to the genome but also are linked to environmental factors. If you just merge them and mix them, the whole data set, you lose effects. And I'm sure this is true for many data. And on top of this, it is always the simple approach that we do as when we do the data analysis to leave the sex chromosomes out, because we say, oh, with this imbalance we have there, it's very hard to normalize. So just use the autosomal genes and see what's the difference. But I'm sure if we do proper analyses and then include the sex chromosomes as well, we will find more differences that we otherwise would miss because we don't have the proper analyses. Or do the proper analyses. And finally, I would like to give you an overview of a current project where we are still working on. This is a sex-specific transcriptomic analysis of a single nuclei RNA-seq data set. So here, single nuclei have been isolated and gene expression has been measured genome-wide for individual cells. So it's a single cell approach. And in this study, there were about 100 donors. This is a post-mortem brain study. So it's not blood, but this here is post-mortem brain of PD patients and controls. And it's quite balanced. So 45 males and 28 female PD patients, 15 male and 9 female controls. Then 5 brain regions have been investigated that are affected in the course of the progression of the disease, also at different time points. And then with the single cell data, all cell types available in the brain, of course, can be profiled. And the first question we ask is if there are any sex-dependent differences in cell type composition of these post-mortem brains. So with this single cell data, you can now for each brain region that has been investigated, ask what is the proportion of cell types we can see in control males, PD males, and so on. And while there are visible differences between the groups, they are minor. They are not strong and not in our analyses, they were not significant. So overall, the differences we see are not on cell type compositional difference, but they must lie in the cells themselves. So we don't need to control extra by cell type composition, but now can go into a cell type specific analysis to really find the gene expression differences. And if you do this in a non-sex stratified way, it's simple. You ask what is up or down regulated in Parkinson versus control. And you can get up or down regulated genes just as I've shown you in this heat map. But if you now want to do this sex stratified, you suddenly have many different patterns that you can expect and that can occur. And this is also a figure from Enrico Glab's summary where he gives an overview for sex-specific or sex-stratified analysis to pinpoint or distinguish all the categories one can identify. So for example, again here if you have control versus patient, control male and female separate, you can either have the scenario where genes are up or down regulated identically between males and females, so we would call this sex neutral, sex independent. You would get these hopefully also if you just throw everything together and do an analysis where sex doesn't play a role. Or you can have these effects that are specific just to one sex. So genes can be upregulated only in female patients, no effect in male patients, or these effects can even be sex dimorphic. You can have downregulation in females and upregulation in males, vice versa. You can have sex-modulated effects where you have the same directionality but on a different magnitude. So suddenly you see if you do this analysis, it gets quite complicated. But I guess we have to do this exercise because if we look at the data sets, we can see that for all brain regions, for all cell types we can look at, we see all these categories. There are sex-neutral genes, there are female-specific, there are male-specific, and there are dimorphic genes that come up in almost every cell type, in almost every brain region. And we wouldn't miss all these effects if we just put everything together. And we wouldn't miss all these effects if we just put everything together and ask where are the differences. But now if we have all these data, it's overwhelming. And how do we make sense out of all these many changes, and what are maybe also false positives, but what is also true? And then you need different approaches to start with this here. The simple approach we first took is to ask what changes are equal between all the cells in all brain regions independent of sex. So the question, what really is Parkinson's disease? What kind of differences do you see independent of sex, independent of the cell type one is investigating? And there are indeed genes like heat shock proteins that come up in all the comparisons. So they are really fundamental to deal also with the overload of protein we have in Parkinson's disease. You can find them everywhere. So you could hopefully also find them if you do a sex-independent analysis. And then you can ask which genes are really region-dependent. Does it make a difference if I look at the prefrontal cortex or at the area more on the brainstem? What is really specific for that region? And you can already see here that there are quite a few region-specific expression changes one can observe. For example, in mitochondrial genes, we focus on astrocytes and just compare two brain regions. You can see here that several of key genes that are linked to mitochondrial function are really dependent. The expression is really dependent on the region you're looking at. And the heat map here also shows this nicely, that in males here you have a way higher level than in females in the prefrontal cortex, but in this region in the brainstem, there's almost no change. So it matters also where you look if you see male or female specific effects. If you then look more closely and have these region-specific effects of mitochondrial genes in astrocytes, you see that most of the effects are actually female-specific. So it's the female where you have the gene expression changes compared to the males. If you plot that as a scatter plot, if the fold changes, if they are similar, they all should be at the diagonal. But if they differ between males and females, they are shifted accordingly. And here you can see it's all shifted up on the y-axis, where you have the female expression changes depicted. And then you can see that here really the major changes can be found in the female astrocytes. If you look at the region-independent effects, what really came out strongly in our analyses were genes that are linked to myelination, and myelination-related genes in oligodendrocytes and in healthy controls. There you have a large number of region-neutral genes that differ in Parkinson's disease and in controls independent of where to look. And if you then consider sex as a variable, you can see a nice sex dimorphic pattern. Here, this scatter plot shows you that there's an upregulation in females, so it's in this direction higher than in males, where on the x-axis we have the difference in male. You can see there's a strong downregulation, so dependent on the sex, but region independently in oligodendrocytes, that we see that myelination really is different. And this has also been shown by Enrico Glab in a study of Alzheimer's disease. So maybe these effects on oligodendrocytes are also apparent across disorders and can explain some of the similar phenotypes we see across neurodegenerative diseases. And with these cool single-cell data, you can now play around and also do cell-cell communication analyses and create these fancy plots that in males or females show you how many interactions you can observe between the different cell types, for example, glutamatergic neurons and GABAergic neurons. And the stronger the color and more lines you see, the stronger the interactions are that are predicted based on these gene expression data. And the only message I want to bring across here without going in detail is the picture looks quite different between males and females. So it's not only differences within the cell, but it's also the communication event between the cell types that really is sex dependent, and where you, for example, here in females, have strong interactions with glutamatergic neurons and oligodendrocyte precursor cells, but this is much weaker in males. So we also then, with these kind of data, can understand better how cells interact with each other in a sex-dependent manner. So I hope I could convince you that in all these analyses, we have to really want to take sex as a biological variable, we have to consider or adapt our study design and our data analysis. It's already important from the beginning when we do our sample collection to really have a sex balanced sample collection to be able to understand the differences. We also should not have one sex collected at one time point and the other later, just to fill in the gap, and then have a bias when we prepare our libraries to be sequenced. We really also should avoid any sex-linked batch confounder effects we have when collecting the data. So the study design here is really important to be clean and balanced to come to conclusions we need to understand these disorders better. And also for the data analysis, we should take sex as a covariate during batch correction, but then really do sex stratified analyses, even if it's more complicated, even if it's overwhelming, even if it leads maybe to false positives, but it's worthwhile following them up and really carefully trying to understand where are true differences between males and females, and how can they also then be used in therapeutic approaches or in diagnostics. As you know, we've been successful to convince the DFG last year to start a priority program based on sex differences in glia cells. Glia cells, because I hope I could convince you, beyond neuronal cells, glia cells really play a key role in several disorders, most likely also in Parkinson's disease. And here in this priority program, we want to combine experts from glial biology who know and study physiology, behavior, brain function, and brain circuits in a sex-specific manner with experts in functional genomics that are good in profiling epigenetic and transcriptional control. Because often our studies remain descriptive. All the analyses I've showed you is nice to have, but it's not validated in any way functionally. It only gives us hints to be further followed up. And this connect and the collaboration with the physiology and the behavior and brain function really tries then to bring together these two layers to understand how the profiles we observe on a descriptive level are linked to functional differences. And of course, we need strong groups that can do proper computational biology and data integration in this process. And we are focusing, or we convinced the DFG that it's worthwhile focusing on glial cells because they are supporting the neurons in several ways. And they are hormone sensitive, maintaining several receptors are important for the homeostasis of neurons. There were really nice studies already coming out, like the one here from Anna Orr on hippocampal astrocytes, where she convincingly also could show differences in glutamatergic receptors linked to differences in sex-specific memory and effects she could observe here on a behavioral level. And if you look in the literature, there are 10 times more studies on sex differences in neurons than sex differences in glia cells. So there's definitely a need to catch up in a coordinated program and bring together data to move the field forward. So we envision in our consortium to have groups that work with astrocytes and oligodendrocytes, so macroglia cells that use human and rodent model systems along with in vitro systems from birth to adulthood to really capture also early and late hormonal influences, and then have this combination of different technologies, the profiling on one hand, but also all these functional approaches to really go beyond description. And we are now at the step where the consortium is evaluating the proposals that have been handed in last year, and we hope to know then in summer who will be in the consortium and can get started in fall. And it would be wonderful if also with this SPP we could connect to your efforts here in Lübeck and to move that further forward, make most out of it. With this, I'm at the end of my talk, and I'm thanking all the collaborators, the financial support, and you for your attention.